Hello, everyone. My name is Andrew Kim, and welcome to the third unit of the AP Bio Review session. Now, the third lesson will be about Unit 3, which is enzyme, cellular respiration, and photosynthesis. Now, these are very complicated concepts, okay? Probably not enzymes. Enzymes are simple, but cellular respiration and photosynthesis happens across a lot of different steps, and each of the steps require a lot of things to remember. So it's going to be a little bit complicated, but I'm going to try to summarize all of them as simple as possible. So let's move on to the actual part. So first of all, enzymes. Now, enzymes are the easiest part of this unit, okay? So enzymes, what is enzyme? What is the definition of an enzyme? Well, enzyme is a catalyst. Now, what does a catalyst mean? Well, we're not talking about catalyst project in SAS, which is my least favorite class this year. Sorry, Mr. Booth. But catalyst that we're talking about over here is something that can catalyze a reaction. Now, what does catalyze mean? It means promotes. It means to support, to make something happen more easily. So when we're talking about when we're in a bio perspective, catalyst means something that makes a certain chemical reaction happen more easily. So the definition of enzyme would be a catalyst that catalyzes chemical reactions by lowering activation energy. Now, what is an activation energy? Activation energy is the least amount of energy required for an actual reaction to happen. So you would often see this graph, time, energy, and it's like this. Now over here, the activation energy is from here all the way down to here, because initially you have this amount of energy, but ultimately you have to have at least this amount of energy in order to really, go over this hump and make this reaction happen. Now, some people might ask me, Andrew, isn't this the activation energy? Because that is the difference in the energy before and after the reaction. No, no, no. Although this is ultimately the difference, still, if you don't have this additional energy, you won't be able to really go over this hump, right? You have to go over the hump in order for the reaction to happen. So this is the activation energy. But obviously, if you look at this graph, the activation energy is too big, so the reaction is not very likely to happen. Now, what enzymes do is enzymes lower the activation energy like this. You can see how the height of the hump is lower than the original graph. This means that the activation energy has decreased, so now you only require less energy for the same reaction to happen, so it is much more likely that the reaction will happen. In other words, it catalyzes the reaction. That is why that is what enzymes are, okay? Now, there are some important things that you have to know. First of all, we have to know how enzymes look like. Now, of course, enzymes will look very complicatedly, but if we simplify that, it looks like this. And then you have something called a substrate. Now, substrate are the actual components that will go through reaction. This is enzyme, this is substrate, okay? Substrate is the reactant. They are the participants of the reaction. Then what is an enzyme? Enzymes never participate in the reaction, but it offers this hub, this place where reaction can happen more easily with lower activation energy. So enzyme, think of enzyme as like Tinder, okay? It's like the hub. It's a platform where reactions can happen, whereas substrates are like people looking for, partners in Tinder, right? They're the actual people who are participating in the reaction. Substrate are like people on Tinder, Tinder users. Enzyme is like Tinder. It's like a hub. It's a platform, okay? So now, as you can see, substrates and enzymes are one-to-one, okay? Enzymes are substrate specific. There might be a substrate that looks like this. There might be a substrate that looks like this. There might be a substrate that looks like this. But this enzyme will only, as you can tell from the shape, take this one specific type of substrate, okay? So enzymes are substrate specific. It can't be used for any substrates, but it can only be used for certain types of substrate. However, there is something called induced fit model. What is an induced fit model? Well, sometimes the shape of the enzyme is slightly off. Let's say the angle of this triangle is slightly smaller. still the enzyme will sort of change its shape by a little bit, so that if there's a small difference in the shape of between the substrate and the enzyme will actually modify its shape a little bit so that the two can combine, and the enzyme can provide a meaningful platform to the substrate so that the substrate can actually go through the reaction at a lower activation energy, okay? So this is induced fit model. You kind of modify the shape a little bit so that substrates can actively be combined to the enzyme, okay? So that is induced fit model. Now, I will drop some important information about enzymes, okay? Three key, 3 stars of enzymes. Just remember these 3, okay? First is called substrate specific. Well, I just mentioned that, right? Enzymes are substrate specific. Enzymes don't belong to the street. They don't fit to anyone. They only fit to specific types of substrates. Second, they're not disposable. When you match a partner on Tinder, are you just gonna delete Tinder? No, of course not. You will use again and again, right? It's not disposable. Enzymes could be used multiple times, but of course, once the substrate gets detached, the next substrate can come in, right? So it's not disposable. Last, they're normally called ACE, synthase, something ACE, whatever ACE. If you have that ACE at the end, then you can tell that it's probably going to be an enzyme, okay? So these are 3 important common sense that you have to really know if you want to get a 5 in AP Bio exam. Now, I will move on to the next important part of enzyme, which is inhibition. Now, what is inhibition? As you can tell from the name, inhibition is blocking, right? So when, let's say you have an enzyme like this, you have a substrate like this, the substrate wants to be combined to the enzyme, but it can't. Why? Because inhibition is happening. Now, inhibition happens when there is a spy. It's James Bond, okay? He does not belong to this enzyme. This chip should not technically combine to the enzyme. It doesn't do anything even if it combines to the enzyme, but it sneaks in and combines. It fills in the spot so that this actual substrate loses the spot to be combined. Do you get what I'm talking about? So it sneaks in, it fills in the spot so that the actual substrates who actually need to go through the reaction cannot go through the reaction. Why? Because again, their platform is already filled up. Now, this type of inhibition is called competitive inhibition. Why? Because it's competition, right, between this black substrate, legit substrate, and blue ripoff substrate. They're competing so that one can fill in the spot of the enzyme. So there's a competition going on, so we call that a competitive inhibition. Now, of course, that means we also have a non-competitive inhibition. Now, this time, enzymes or fake enzyme and enzymes aren't competing for the same spot, but rather you have a legit substrate that wants to be combined, but then there's this spy that does not fill up the same spot. So this time, this spy is not competing. This is not happening, but rather it sticks to a different part of the enzyme and it modifies the shape of the enzyme. Denaturation happens. The enzyme shape will change like this. Oh no, now since the shape is all changed, this enzyme, this substrate cannot fit into the enzyme because the shape of the enzyme has changed. Now again, these two guys aren't competing for the same spot. That is why it's not competitive inhibition, but still inhibition is happening because the blue chip is changing the shape of the enzyme so that the substrate cannot fit into the enzyme, okay? So these are 2 different types of inhibition. And last is called something called feedback inhibition. Now, what's interesting is if enzyme and substrates just continue combining and reaction happens and a product is made, so many products, oh my gosh, you will have too much of these products, right? The concentration of this product will increase too much, which will be bad for your body because your body health is entirely dependent on something called homeostasis, right? It has to be stable. But once you continue just making these products through the reaction caused by the enzyme, your body's not going to stay healthy. That is why there's something called feedback inhibition. Now, feedback inhibition is you make this first product, right? Through the reaction. Then this product, let's say this red product is made out of the reaction between enzyme 1, substrate 1, okay? Now then, this product itself can also act as a substrate for a different enzyme, enzyme 2, substrate 2. Now then, through the reaction between these two, another product is made. This time, this product is blue and it's called product 3. Now, product 3 can also work as a substrate by itself, and it gets combined to a different enzyme, enzyme 3, substrate 3. Now, these, again, go through a reaction and will make a new chemical, which can also be a substrate, and let's call this substrate 4. Now, what's interesting about substrate 4 is this substrate 4 can combine all the way back to the very first enzyme that we started from. Now, what happens? Competition happens. Between number 4 and number 1 substrate, number 4 substrates will take some of number 1 substrate spots, right? Some of the number 4s will fit into enzyme number 1. What's going to happen is enzyme substrate number 1 will decrease when it comes to combining to enzyme 1, and so the reaction will stop. Why? Because again, competitive inhibition is happening. Through this process, we can regulate the production of each enzyme, right? Because or else we would just be continuing making enzyme 1 makes enzyme 2, enzyme 2 makes enzyme 3. Enzymes are substrates are continuously made, but through this sort of a break, this competition, this reaction, this chain will reduce its speed. That's what we're talking about in feedback inhibition, okay? Now let's move on to the next important part of enzyme, which is denaturation. I mean, I just talked about it, right? Denaturation is when the enzyme changes its shapes. Now, I explained this in unit 1, right? Proteins go through denaturation when pH, temperature, or salinity changes. Enzyme is a type of protein. Enzyme is a type of protein, okay? Now, you will learn this. I'm pretty sure you learned this in pre-calculus. It's a sign you use in set theory, but whatever. Enzyme is a part of protein. Enzyme is a type of protein, so enzymes do also go through denaturation, okay? Next important is cofactor and coenzyme. Now, not very important, but sometimes, let's say you have enzyme 1 and enzyme and substrate 1. Now, as you can see, substrate 1 is too narrow to fit in enzyme 1, right? Then sometimes there are coenzymes. Enzyme 2, let's call it. Sorry, cofactor, let's call it substrate 2. Now, with these two pieces, they can actually fit in this spot, right? So sometimes not only one substrate, but multiple substrates can combine together into this enzyme as a cofactor, okay? That can also happen, but I never saw this in APTQ or whatever quizzes, tests, so probably don't really need to know, but I mean, there's nothing to lose, right? So let's just keep this in our mind. Last is concentration of substrate. Now, again, let's bring back the Tinder analogy. If we have, let's say, a lot of Tinder platforms, but you only have one user, then only one enzyme will actually be combined to the substrate and only one reaction will happen, right? But of course, if we have more Tinder users, then more Tinder users will match up with each other and more reactions will happen, right? So of course, when the concentration of a substrate or the Tinder users increase, the number of matchups or the number of reactions will increase. However, if we have too many substrates, if we have too many Tinder users, then of course there will be some jams, right? On the network. Of course, not everyone, even if there are 10,000 people, 100,000, 100 million people using Tinder, still there will be a limit. That is why as the number of substrate increases, the reaction speed will increase initially, but it will die off at a certain level. Why? Because if every single Tinder platform is occupied by a substrate, then even if you have these extra substrates, where will they meet up? They won't have any platforms left. That is why initially, until the platforms are all occupied, there will be an increase in reaction speed as substrate concentration increases. But once all the platforms, all the enzymes are occupied, the speed will die off. Now that is it for enzymes. And now I will move on to the cellular respiration part. Okay. Now, this is where things get kind of difficult, okay? So if you need some break, then go get the water, use the bathroom or whatever and come back, okay? Cellular respiration. Now, oftentimes people think cellular respiration is a process of destroying energy. You're just, you're getting rid of all the energy. That is wrong. As I explained in the previous unit, energy cannot be made nor destroyed, right? It's just converted from one type to another. And in cellular respiration, we're changing chemical energy into usable energy. In other words, ATP. Okay? So cellular respiration is all about changing the chemical energy stored in glycogen, glucose, whatever carbohydrates into a usable energy so that you can actually throw your fist, so that you can flex your muscles, so that you can think, so that you can breathe, so that you can do whatever thing, okay? So now, let me explain how that actually happens. First of all, cellular respiration, as I explained in the previous video, happens in mitochondria. But before that, I have to explain that cellular respiration happens through two different types of ways. First is with air, second is without air. Now, with air is more difficult, and that is why I will explain with air first, because once we understand with air or aerobic respiration, anaerobic or no air respiration will come more naturally, okay? So let's first get to know about aerobic or with air cellular respiration first, okay? So now, I mean, intuitively speaking, with air makes more sense, right? Because at the end of the day, cellular respiration is sort of like breathing. So of course, with air sounds more natural, but I'll show you how that happens. So number one, aerobic. Now, in aerobic respiration, there are three big parts and one intermediate part, okay? So first step of aerobic respiration out of the three steps is called glycolysis. Second step is called Krebs cycle. And last is called ETC, or electron transport chain. And in the middle, you do have an intermediate step. So let's see, let's have a look. So now glycolysis, as I've explained, happens where? Well, it happens in mitochondria, but glycolysis is the only step that happens outside, right outside of the mitochondria. It happens in the cytoplasm. So as you remember, if you have a cell, if you have nucleus, if you have all the aparatus, whatever thing, Golgi apparatus, whatever things, then you have a mitochondria here, right? Then right outside of mitochondria, this is where cytoplasm is, right? And this is also where glycolysis will happen. Now, what's going to happen is when you have a glucose, this glucose will first split, change its format to a different type of carb, but you don't have to remember what it is. And then it ultimately splits into two pyruvates. Pyruvate 1, pyruvate 2, okay? So this is the first step, okay? Glucose, which is sugar stored in my body. You might ask, Andrew, what do you mean I have sugar stored in my body? When I lick my body, it's not sugary, it's not sweet. I don't have any sugar stored in my body. No, you're wrong. Carbs, whenever you eat carbs, and specifically we're talking about plants over here, right? So especially for plants, they would store all the energy in the form of sugar or glucose, which is C6H12O6. Of course, you won't be able to taste it, but it exists in your body, okay? So now glucose, C6H12O6, splits into two pyruvates. Now let's think about it. How many carbons would each pyruvate have? 3. Why? you had 6 carbons in glucose, and if that splits into 2 equal parts, if, of course, each pyruvate would have 3 carbons, right? This is not very important, but just to help your understanding, right? Now, what's interesting is on the first change from glucose to a different type of carb, what's going to happen is 2 ATP will change to 2 ADP. Now you might ask me, Andrew, this is weird. I thought cellular respiration is all about creating ATP. Why are we changing ATP back to ADP? Well, this is for later usage. Don't worry. Yes, we are losing 2 ATP right now, but later we will bring a comeback and we will make more ATP, okay? And then when this carb splits into 2 pyruvates, for each pyruvate, 2 ADP changes to 2 ATP, and 1 NAD plus changes to NADH. Since you have 2 pyruvates, ultimately, you will have 4 ATP made minus 2 ATP, right? Because you're splitting 2 ATP over here, and you would have 2 NADH made. So ultimately, after this first step, which is glycolysis, you will have 2 ATP and 2 NADH made. Now, as I told you, now we have actually positive net gain of ATP, right? Because yes, we did lose 2 ATP here, but per pyruvate, we're gaining 2 ATP. So ultimately, we're gaining 4 ATP. So we have 2 ATP gains. Now you might ask me, Andrew, so then what is NADH? NADH is an electron carrier. NAD plus is like an empty carrier. NADH, by adding that H and electron, is a carrier with your electron sitting on it. Now, why are we making these electron carriers? Well, these will be sent all the way down to ETC, electron transport chain, for further usage. So for now, just know that, oh, you make ATP out of glycolysis, and you also make NADH, which carries electrons for further usage, okay? Now, intermediate step, not very important, but just know that in the intermediate step, the pyruvate, because we created 2 pyruvates up in glycolysis, right, will change to something called acetyl-CoA. Now, again, you don't really have to remember all these names, pyruvates, acetyl-CoA, but just remember how this process happens. In glycolysis, it happens where it happens in cytoplasm. It makes pyruvates, which makes ATP and electron carrier, okay? That's the main takeaway. Now, in this process, CoA will change to CO2. And again, NAD plus will change to NADH. As you can see, more electron carriers are made. And also one interesting thing is CO2 is made. Of course, in cellular respiration, the product is CO2, right? So it is very natural that CO2 is made. Now, before I finish explaining cellular respiration, I'll write the chemical formula, chemical reaction for cellular respiration. That is C6H12O6 plus CO2 becomes H2O and O2. Now, I did not balance this out because I forgot the I think I made a small mistake over here. Yep, this is the good old cellular respiration formula. I'm pretty sure you guys have seen this somewhere. It's very common, right? Yeah, I think I balanced it correctly. So this is a reaction for cellular respiration. As you can see, 1 glucose, 1 sugar, and a ton of oxygen is made to create H2O water and CO2 carbon dioxide, right? So as you can see over here, CO2 carbon dioxide is made as a product, right? So we can really kind of see where we're going, okay? So ultimately, we want to have this, okay? Now, and we will have some ATP made. So as you can see, some ATPs are being made and some CO2s are being made, right? Now, Krebs cycle is the most complicated part of cellular respiration, but what you really just have to know is this acetyl-CoA made in the intermediate step will go into this cycle in matrix, okay? So these 2 steps, glycolysis and the intermediate step, happens in cytoplasm. So right outside of the mitochondria, but the Krebs cycle actually happens in the matrix of the mitochondria. And matrix is here. Okay, so it actually happens in the matrix. Now, Krebs cycle, again, acetyl-CoA, which is the product of the intermediate step, will go through this cycle where ultimately 3NAD plus will be converted to 3NADH. One FAD will convert to FADH2, and ADP will be converted to ATP. Okay, so going through this cycle, okay, it's kind of like when you're playing baseball, you have all the batting cycle, right? Going through the cycle, you have some amount of hits, some amount of doubles. If you're lucky, probably a home run, right? Similar to that, going through the cycle, you'll make these reactions happen. So again, NAD pluses will be converted to NADH, meaning that empty electron carriers will actually start carrying electrons. FAD is a different type of electron carrier. So again, there's another empty electron carrier that will actually be filled with an electron. And again, ADP will be converted to ATP. Now, as you can see, we have ATP being made here. We have ATP being made here as well. But still, that is not enough ATP, right? We want to make more ATP. And that is why we had all these electron carriers, okay? Now that all these electron carriers are actually carrying electrons, these electron carriers will bring electrons to ETC. And where does ETC happen? In mitochondria, you remember this place, this borderline between the intermembrane space and matrix. This borderline is where ETC happens. All these electron carriers that are actually carrying electron will bring electron to this border so that ETC, the last step, can happen. The last step is where all the ATPs, well, not all, but majority, a vast majority of ATPs will be made. And I'll explain how that happens. Now, let's zoom into this borderline. Okay, let's zoom in. Now, how this looks like is you have this border and you have a lot of proteins, a lot of synthases, okay? And as I have mentioned, there will be electrons being delivered to ETC, right? For example, NADH or FADH2, right? Now, what these electron carriers will do now here is they will give out the electrons, and these electrons will go through this chain. That is why it's called electron transport chain. Electrons will flow through this chain like a wave. It's going to flow through it. And what's going to happen is as the electron flows, similar to a tide, it's going to create small energy, which makes H pluses from this side, which is matrix, right? Because we're in the matrix here, and this will be the intermembrane space. H pluses will move out of the matrix and move to the intermembrane space. Do we understand? Okay. As electron flows, there's a small energy generated. Using that energy, A pluses will move upward. Ultimately, we will have a lot of H pluses on the intermembrane space, but very few H pluses actually left in the matrix, meaning that there will be a gradient made. High concentration, low concentration. Okay? Now, what's going to happen is since the concentration of H plus in the intermembrane space is higher, all those H pluses will move down. Why? Because it's higher concentration. Diffusion always happens from higher concentration to lower concentration. So all the H pluses will flow down, and using that force of H plus flowing down, we will generate energy in this synthase to convert ADP to ATP. This is where majority of the ATPs are created. It's kind of like those wind power generators, right? As wind flows, as a turbine turns, energy is created, right? Similar to that, here, instead of wind flowing, H plus will flow through the ATP synthase, and through that force of H plus flowing, there's going to be like a small turbine. Of course, there isn't an actual turbine, but it's just like how there's a turbine, energy is going to be generated, and using that energy, ADP will be converted to finally ATP. Now, then the last question is, what about the electron? What about the E minus, this electron that traveled through this chain, which enabled H pluses to go up? What's going to happen to this electron? Of course, we can't just make these electrons be left in our body. Then we will have too many E minuses, electrons left in our body, right? That's going to be toxic. What's going to happen over here is O2 will come up. It's going to combine with E minus and finally turn into H2O. That is why O2 is required in cellular respiration. Without O2, these used E minuses, electrons, will be left in our body and ultimately kill us. But if we have O2, these O2 will pick up E minus. You're used, electron, come over here. You're already used. I'll pick you up and combine to you so that now we become a different species, which is A2O. And that is again why H2O was the product, the end result of cellular respiration. And that is pretty much it for the aerobic respiration. So it was very complicated, but just to summarize very simply, just remember this. We have 3 steps. In first 2 steps, we will mainly convert NAD plus to NADH. Why? Because we want to make the electron carriers actually carry electron. The first 2 steps, glycolysis and Krebs cycle, is mainly about shoving electrons onto electron carriers. Of course, we will make a couple of ATPs, but not a lot. And once we make all the electron carriers carry electron, these electron carriers will shift, will move to ETC, where they will release electrons, and those electrons will be used to go through the chain to pump H plus to the other side to make a concentrated H plus in the intermembrane space, so that there's a gradient made so that the H plus can flow back down. And using the force of H plus flowing back down, we can generate ATP. And again, this used up E minus electron will be picked up by O2 and H plus to create H2O. That is pretty much it when it comes to cellular respiration. Of course, it's better if you remember all the pyruvate, ATP, ADP, CO2, acetyl-CoA, CoA, and everything, but not very necessary. Just remember this whole big picture. And the smaller details, you can probably figure it out in the testing spot when you're taking the exam, actually. Now let me move on to the next part of cellular respiration, which is, again, anaerobic respiration, which is, as you can tell from the name, no air respiration. Now, this sounds weird because I just mentioned how O2 is important in respiration, because O2 picks up the wasted, the used electrons, so the electrons don't just stack up in our body and ultimately kill us. But that is why anaerobic respiration does not go through ETC, because yes, ETC requires O2, but the first 2 steps don't, right? So anaerobic respiration will only go through glycolysis initially, okay? So glycolysis. Now, what happens in glycolysis again? Glucose or sugar will be break down into 2 pyruvates, right? In that process, I mentioned how 2 ATP is made and 2 NADH, electron carriers, will carry electron, right? This was the first step of aerobic respiration. Anaerobic respiration and aerobic respiration will share the very first step, which is glycolysis, right? Now, once glycolysis happens, now the issue is, well, the good part is, first of all, we have ATP made. Yay, ATP, cool. I mean, the whole purpose of respiration is to create ATP, and we have ATP created, so isn't that pretty much it? I mean, it is good, but the issue is that we have NADH, and NADH, when it gets stacked in our body, can also be very harmful. So we have to release NADH. And how we release that is we go through something called fermentation. okay? Now, we have two different types of fermentation. First is alcoholic fermentation. Now, as I've mentioned, pyruvates were created, right? This pyruvate in alcoholic fermentation will change to alcohol or ethanol. And in that process, NADH will be converted to NAD plus. Yay, we have NADH, which can be toxic to your body, eliminated, and NAD plus made, right? So now we don't have the toxic thing left in our body. That is why this is a more sustainable way, right? Second part of fermentation is called lactic acid fermentation. Sorry, not second part, this is the second type of fermentation. So again, if you have pyruvate made, then this pyruvate will change to lactate. And in that reaction, again, NADH, which is toxic to our body, will be converted to NAD plus. You can see how both fermentations purpose is literally the same. It just wants to get rid of NADH because NADH is toxic to your body. So just remember, anaerobic respiration is essentially the first step of aerobic respiration, which is glycolysis, changing glucose to pyruvate. And in that process, we make ATP, which is the whole purpose of respiration. The issue is, while ATP is made, NADH is also made. And since NADH is toxic, we have to get rid of NADH. And how we do that is we do that through fermentation. And there are two different types of fermentation. First is alcoholic fermentation. Second is lactic acid fermentation. And both fermentation, what they do is they change pyruvate to something else, rather that's ethanol or lactate. And in that process of changing, you also convert NADH into NAD plus, which is less toxic to your body. That is it for anaerobic and aerobic respiration. Great, great job. It's really difficult, but you made it. Now, finally, we have one last thing left, which is photosynthesis. Photosynthesis. Now, photosynthesis, where does photosynthesis happen? Well, as I explained in the last video, it happens in the chloroplast, right? Now, we have to first examine the structure of chloroplast. Chloroplasts, as you guys would know, have double membrane, and inside there you have something called thylakoid. And this empty part, this vacant part that is not thylakoid, is called stroma. Okay? Now, photosynthesis happens across two different steps. First is something called light-dependent step. And the next step is called Calvin cycle. Both needs to happen together, okay? Light-dependent cycle itself would not solve the purpose of photosynthesis, and Calvin cycle cannot just happen by itself. Both should happen together, just like how in aerobic respiration, you needed all three steps, right? So now, before I explain the nitty-gritty parts of photosynthesis, let me give you a big picture of what photosynthesis is. Again, photosynthesis is the opposite of cellular respiration. Cellular respiration was a process of converting chemical energy stored in your glucose, your sugar, to usable energy, ATP. Photosynthesis is the exact opposite. It is converting ATP, or it is converting sunlight into chemical energy stored in sugar. Okay, so photosynthesis changes sunlight to sugar. This is photosynthesis. And cellular respiration is changing sugar to a usable energy, which is ATP. Okay, and the chemical formula for photosynthesis will be H2O plus CO2 makes C6H12O6, which is sugar, plus O2. Okay, now let me first explain the light-dependent process. Light-dependent step. Now, light-dependent reaction happens in thylakoid, okay? So let me zoom into thylakoid. Let me draw a bigger thylakoid. Now, if you have a thylakoid like this, first of all, again, H2O is the reactant of this reaction, right? It's what is used in this reaction. It's an ingredient. So H2O comes in, which is water, and it will split into three parts: electron, H plus, and O2. Now, very similar to electron transport chain that we observed in cellular respiration, this electron will go through this chain. And when it goes through the chain on thylakoid this time, not in mitochondria, similarly, small amount of energy will be generated, which will move H plus is created here to move upward and outside of the thylakoid and to the stroma. Now, what's going to happen is, again, very similarly, you will have so many H pluses outside of the thylakoid. Why? Because you're pumping out all the H pluses outside using the small energy generated by electron transporting. So you will have high concentration here and low concentration in the thylakoid. Now, again, what's going to happen is all the H pluses will move back into the thylakoid, which will create ATP. So essentially, exactly the same thing as ETC, electron transport chain of cellular respiration, is happening here. Now, then you might ask me, why are you creating ATP? I thought you wanted to create C6H12O6 or glucose or sugar. Well, that is because ATP is the ingredient for Calvin cycle. In Calvin cycle, you will convert ATP to sugar, and I'll show you how that happens. Now, let's talk about Calvin cycle. In Calvin cycle, essentially, it's very simple. Calvin cycle happens in the stroma, okay? So once ATP is created through light-dependent process, this ATP will move to stroma where Calvin cycle happens. And in Calvin cycle, CO2 will be converted to glucose. And in that process, ATP will turn into ADP. ATP enables CO2 to be changed to glucose. And finally, that is how we have glucose or sugar being made. Now, Calvin cycle is very simple, right? It's a simple process. What's important to remember in photosynthesis is it has two steps, and first step is very similar to ETC. Its purpose is to make ATP, and that ATP created in the first step will be used in the second step in the stroma to convert CO2 to sugar. Okay, so that is how photosynthesis works. There is a reason why I explain aerobic respiration the first, because once you understand aerobic respiration, anaerobic respiration, photosynthesis, they're all very similar, right? So as that was pretty much it for cellular respiration, photosynthesis, and enzyme, and the entirety of Unit 3. Thank you so much for watching this video, and most importantly, don't forget to have a great day.